What's up, guys? Dr. Serop here. As you can see, I'm joining you from outer space. Check out this shooting star. Pretty cool, right? Just kidding. Let's erase this. All right, so today we're going to talk about the neuromuscular junction, otherwise known as where the nerves meet the muscle. So, before we start talking about the actual anatomy and physiology of the neuromuscular junction, let's talk about some disorders that occur at the neuromuscular junction. The first one I want to talk to you about is something you might have heard of before. It's called multiple sclerosis, or MS. The second one is actually something we have turned into a type of therapy. It's called botulinum toxin, or Botox. We'll talk about Botox in a bit. Number three is something you probably haven't heard of, but maybe you have. It's called myasthenia gravis. Number four, you likely have heard of before. It's called muscular dystrophy. And the last one, kind of weird to put on this list because it doesn't happen in live humans. It happens when you die, and that's called rigor mortis. So, the neuromuscular junction, if you break that word down, stands for where the neuron meets the muscle. So what we're going to do now is draw where the neuron is meeting the muscle. So this blue part here is representing the nerve. And if you've done some basic anatomy on nerves, you would know that the long tail of it is called an axon. This red part here, and let's erase this so you can see my face better. This red part is the muscle, and the membrane of a muscle is called the sarcolemma. The space in between the neuron and the muscle is called a synapse. Let's talk about what happens first. The first thing that happens is your brain tells your muscles to contract. If I want to pick something up, it requires my bicep to flex, right? And that flexes the elbow. So my brain tells my bicep to contract, and it does that using a nerve. That signal travels down several nerves in your body, and it reaches this last one that's going to finally talk to the muscle. And the way it does that is sending a signal by depolarizing this axon. You might have heard of a term called a depolarization. And depolarization is when sodium ions are going inside of that axon all along the way. It's depolarizing and making that axon more positive, sending a action potential down to the axon terminal, which is the last part of that axon. So we get traveling of this signal, or depolarization of this axon, all the way down to the end. Now, a lot of nerves in your body have something that is called myelin. Myelin is this fatty covering around the axon that actually insulates that tail and allows the signal to travel faster. That's going to be way important later. So finally, this depolarization reaches the axon terminal, and what that does is it causes voltage-gated calcium channels to open. So here's some calcium. And when those voltage-gated calcium channels open, due to the depolarization reaching the end, calcium then influxes into the axon terminal. What is that going to do? Well, in this axon terminal, we have vesicles filled with a neurotransmitter that's called acetylcholine. When calcium influxes into that axon terminal, these vesicles filled with acetylcholine will bind to the edge of this axon and dump out acetylcholine into the synapse. So now we have a synapse filled with acetylcholine. Acetylcholine is the main neurotransmitter that's going to stimulate this muscle. How is it going to do that? Well, on this muscle, there's this part right here that's kind of dipping in. That's called the motor end plate. On there, we have receptors. And with every receptor, just like a lock and key, that receptor needs something to bind to it. And in this case, it's going to be acetylcholine binding at these receptors. When acetylcholine binds to these receptors on the sarcolemma of the muscle, that's going to cause the sarcolemma to depolarize. Just like this axon depolarized, the sarcolemma will depolarize. And what happens with depolarization? We get sodium, just like up here, rushing in. When positively charged sodium, let's add that there, positive charges. When positively charged sodium rushes into the muscle sarcolemma, that's going to create a positive membrane potential. And it's going to create a stimulus all across that sarcolemma, depolarizing it. So you're actually going to get this depolarization all across the muscle membrane. Because one of the features of muscle is that it's excitable. So if you excite one part of it, the rest of it will excite. Just like if lightning were to hit your pool or a body of water, if you're anywhere inside that body of water, you will get electrocuted because it's excitable. So this sarcolemma then depolarizes, and that action potential goes all the way across that sarcolemma. Now we need to add some structures that are present within the muscle itself. The structures I'm going to draw now are called the T-tubules and the sarcoplasmic reticulum. All right, I've drawn a lot here, kind of this spider web looking thingy. Do you see these two lines that I draw here and here? Those are called T-tubules. T-tubules help to bring that depolarization of the sarcolemma deep inside the muscle. All right, so we had that excitation on the membrane, but if it wasn't for the T-tubules, it would stay on the membrane. But these T-tubules allow that depolarization to travel deep inside the muscle. So now this action potential can go inside the muscle deep where all the work's going to happen. Next is this part that I drew in the middle here. This, in reality, kind of looks like this web, and that is called a sarcoplasmic reticulum. Sarcoplasmic reticulum is where calcium is stored inside of your muscles. So now we have this sarcoplasmic reticulum filled with calcium. It is the calcium storage sac. So we have sent the action potential with the nerve, released acetylcholine, stimulated acetylcholine receptors on the cell, causing sodium to rush in, and that electricity or that depolarization now goes deep inside of the cell. How is this going to make the muscle contract? Inside of your muscle, there are several proteins that work together to make a muscle contract. Now there's going to be, this next thing I'm drawing, there's going to be thousands of these, but I'm going to draw one for the sake of understanding. What we're going to draw right now is the functional unit of the muscle called a sarcomere. This is a sarcomere. The definition of a sarcomere is from one Z line to another Z line, okay? Sometimes called Z discs. And it's called that because it has this Z zigzagging shape. So this Z disc to Z disc is the functional unit of a muscle. That is what's going to get shorter, causing a muscle to contract. Attached to this sarcomere, we have the two proteins that do all the work. And those proteins are called the myofilaments, or actin and myosin. All right, our drawing is getting really busy now. So again, sarcomere is one Z disc to another Z disc. Attached to those Z discs, you have something called the thin filament, or actin. Actin goes along that Z disc, all along it, on every Z disc, stretching. Now, in between those actin, there is the thick filament called the myosin. Now, I've heard this referred to as the love story between Romeo and Juliet. If you remember in Romeo and Juliet were very in love, but they had something stopping them, something getting in their way, and that was their parents, who didn't want that relationship to work. In this case, myosin, or the thick filament, is Romeo, and actin is the thin filament, or Juliet. Do you see how these myosins have these little extending heads coming off, almost looking like a golf club? Those myosin heads really want to bind with actin. So Romeo really wants to bind with Juliet. When they bind to that actin, they bind and then attach and then pull those actins together. And what that does is it causes these two Z discs to come closer, as in the sarcomere gets shorter. And that's the final goal here, is to make that sarcomere shorter. And that's how you get muscle contraction. Now, there's a problem. How do we get this myosin head to attach to that actin? Just like in Romeo and Juliet, it's not as easy as it looks. There's something standing in the way. On this actin, you have two proteins that are standing in the way of that occurring. And those two proteins are called tropomyosin and troponin. Do you see this stringy looking thing? That stringy looking thing is called tropomyosin, and it wraps around that actin. Attached to that tropomyosin is another protein called troponin. Tropomyosin and troponin are like Juliet's parents, trying to stop this relationship from happening. What they're doing is they're blocking an active site where that myosin head can attach. So actin has this active site, almost like a receptor, for the myosin head to bind to so that we can get that contraction. But tropomyosin and troponin are blocking it. Now, what is going to convince them to move out of the way so that myosin and actin can bind? Well, kind of like our everyday lives, somebody cool comes around. Maybe Juliet's cool uncle comes and convinces the parents to let them have a relationship. In this case, that cool uncle is called calcium. Calcium, when the action potential comes from the sarcolemma and down these T tubules, when they depolarize, that causes extreme calcium efflux, and that calcium exits the sarcoplasmic reticulum into the cytoplasm of the muscle. And what is that calcium going to do? What does the cool uncle do? That calcium is going to come and bind to this troponin. And when it binds to that troponin, it's going to move that tropomyosin string out of the way. When that tropomyosin string moves out of the way, these myosin heads can finally bind to actin. And we get our fairy tale ending where myosin and actin have finally bound. Now, what does it require for this to happen? Well, one thing is ATP, adenosine triphosphate, right? Our source of energy for our body. ATP will help these myosin heads attach. What it actually does is it puts the myosin head into this position that has potential energy. It's called the cocking position. And so now we bound that myosin to the actin and we put it in a cocking position ready for what's called a power stroke. And those myosin heads can perform this power stroke and pull on that actin, pulling the Z discs together. Those Z discs shorten the sarcomere all along the muscle. Several thousand sarcomeres get short and the muscle can finally contract. Then these myosin heads need to detach. They detach from actin with another ATP. So ATP is needed for both contraction and relaxation. What happens is the myosin heads grab the actin, do the power stroke, let go further down the actin, grab on, power stroke, let go, further down, grab on, power stroke, let go, and this muscle gets shorter and shorter and shorter. And then finally, when you with your brain, tell your muscle to stop contracting, then ATP binds and myosin completely let go and that muscle can relax. That is called the sliding filament theory. The way that actin and myosin need to bind where troponin and tropomyosin are standing in the way. Don't forget how important calcium is in order to make that happen. Now, let's go to some of these disorders we talked about at the beginning. You might have heard of MS before. MS, multiple sclerosis, is an autoimmune disorder. What does autoimmune mean? Autoimmune means that the body's own white blood cells are attacking the body. So your white blood cells, which are normally made to fight bacteria and viruses, are attacking things that they shouldn't be attacking. In this case, with MS, your own white blood cells will be attacking your myelin sheaths. And what it does is it starts destroying that insulation on the myelin. And if you remember, what did myelin do? Myelin helped to make that action potential move faster. And if it doesn't move faster, if it starts moving slowly, then you get MS symptoms. Things like extreme weakness in your muscles, and it causes vision issues, and it causes numbness or tingling. So MS is an autoimmune disorder that attacks the myelin. Now, it usually attacks the myelin that is in the central nervous system, which is the brain and the spinal cord, but some evidence is showing that it can attack the myelin in the peripheral nervous system, which is what we've drawn here, close to the muscles. The next disorder that we talked about, which actually now has, funnily enough, become a treatment, is botulinum toxin, or Botox. You've heard of Botox for aesthetic purposes before, getting Botox injections in the face, or, really interesting, getting Botox injections to help prevent migraines. What does botulinum toxin do? By the way, botulinum is a bacteria and it produces a toxin, and we harvest that toxin and use it as this treatment. But if you were to actually get infected with botulinum, or even if you took Botox, how does it work? Botulinum toxin travels and it prevents the exocytosis of this acetylcholine here. So remember how acetylcholine that was inside these vesicles, in response to calcium influx, that acetylcholine was supposed to enter the synapse to stimulate this muscle. Botox stops that from happening. So what would happen if acetylcholine didn't get exocytosed from this synaptic terminal? We wouldn't have any acetylcholine in this synapse, therefore no stimulation of a muscle, otherwise known as paralysis. Botox paralyzes muscles. And that's why it can actually help with migraines. If a patient's migraines are due to tension in muscles, it relaxes or paralyzes those muscles. For aesthetic purposes, they inject it into muscles to relax those muscles, which those muscles pull on the skin and cause those fine lines, right? So if we relax the muscles, then it relaxes the skin. And you don't get that pulling of the skin, and you get that less wrinkled look, okay? But don't forget, it is a toxin from a bacteria. The third one we have listed here is myasthenia gravis. Myasthenia gravis, similar to multiple sclerosis, is an autoimmune disorder where the body's white blood cells attack the body itself. In this case, in myasthenia gravis, the body's white blood cells come and attack the acetylcholine receptors, and they start knocking out our acetylcholine receptors. It actually ends up having the same effect as Botox, where acetylcholine receptors are gone, so acetylcholine has nowhere to bind and it just stays in the synapse. If we don't get acetylcholine binding, then we don't get muscular contraction, and therefore we get that weakness and paralysis of muscles. A lot of these disorders occur where it's okay for a certain portion of the life, but then when it starts affecting vital muscles, tell me, what do you think a vital muscle is? The heart? How about the diaphragm that helps to breathe in and inhale air? When it starts to affect vital organs, that's what actually ends up leading patients to death, because the muscles that are vital, right, not skeletal, but vital muscles like cardiac muscle and the diaphragm fail due to these mechanisms that we're talking about. The fourth one that we have written here is muscular dystrophy. Muscular dystrophy is something that happens in children, most commonly in boys. And it affects, what it does is it mutates a gene called dystrophin. Dystrophin is a gene that binds actin to the sarcolemma. So what you get is this protein right here binding actin to the sarcolemma of a muscle. And what it does is it helps control that muscular contraction. It helps to secure and anchor this sarcomere in place. But people with muscular dystrophy, okay, boys that have muscular dystrophy, they have abnormal dystrophin proteins. And so when that dystrophin protein is abnormal, their sarcomeres aren't secure and they get extreme muscle weakness. They have difficulty with their gait and difficulty walking. And then usually in adolescent or later years, in their 20s or 30s, it can lead to death due to respiratory failure. They're not able to use the muscles to breathe in. The last one here is rigor mortis. Rigor mortis doesn't happen when you're alive. It happens after death. And what do we know after death? We know that after death, you stop producing energy. You stop producing ATP. If we don't have ATP, what happens? We talked about how ATP is needed for both muscular contraction and relaxation. If muscles can't relax due to the lack of ATP, then they will just stay contracted forever. Again, ATP is needed for relaxation. So if we don't make any more ATP, such as after death, those muscles stay contracted and you get rigor mortis, which is all the muscles of the body being contracted. The peak of rigor mortis is usually around 12 hours after death. You'll find the body being very stiff. But after a couple days, these proteins start to just break down in general. The myosin and actin and the cross bridge formed between them just break down. Those proteins denature and then the muscles relax. I hope that the neuromuscular junction and the relationship between nerves and muscles makes a little more sense now. And I hope that knowing that normal anatomy can help you understand these disorders that you've definitely heard of before. Thanks for listening. Can't wait to see you on the next one. Take care.